What if a single gene could flip a switch in your cells, telling them exactly where and how to rebuild a lost limb, fingertip by fingertip? When an axolotl loses a limb, cells near the wound switch back to a simpler state and cluster under the new skin in a bud called the blastema. These starter cells then multiply and turn into bone, muscle, nerves, and skin, rebuilding the limb exactly as it was before. What if we could copy the axolotl's regenerating capability? That's the question 5 researchers asked. Could copying the axolotl's mechanism of regeneration allow us to restore limbs? Just last week, the team unveiled their paper, Molecular Basis of Positional Memory in Limb Regeneration. Scientists have wrestled with decoding how to regenerate complex limbs for centuries, from Aristotle's first biological observations to the salamander blastema cell characterization in the late 20th century. Yet today, limb regeneration remains a major hurdle for humans, limiting their ability to cure effectively. So the researchers started by extracting the cells from an axolotl limb and classified them as front, anterior, and back, posterior of the wound. They discovered that a gene called Hand2 flips on a protein signal called sonic hedgehog, which acts like an instruction messenger telling cells how to rebuild lost limbs. This Hand2 gene is also present in humans. But what was fascinating is that when they inactivated the Hand2 gene, the axolotls were not able to rebuild the lost limbs. Even crazier, when they forced the activation of the Hand2 gene in the front side cells, new extra limbs began to sprout. Now, I think that this paper is one step closer to achieving full limb regeneration in humans. It would be cool to see if this Hand2 sonic hedgehog mechanism uncovered in axolotls also operates in mice. All right, and now the meme of the week. Thanks for watching. If you enjoyed this deep dive into limb regeneration, hit subscribe and check out our video on how we can improve bone regeneration.